Hello. Today we're going to enter into section 9 and we will probably get through section 10 as well. And the last sections of this Fundamentals in Pharmacology course is going to focus on drug development. And the primary goal of this section of the course is to give you an overview of drug development, have you understand the different phases of preclinical and clinical drug development, but most importantly, to try and give some substance to what we've thought previously. So what I'd really like you to do is to apply the knowledge from the previous classes that focused on pharmacokinetics and pharmacodynamics to the process of drug development, because that's really where all of these concepts derive from. So hopefully these sections will allow you to cement your knowledge of the PK and PD concepts that we taught previously. Okay, so when we approach the whole schema of developing a new drug entity, we like to compartmentalize things. So we have three different components. One is the discovery phase. The second is the preclinical drug development phase, or the animal testing phase. And the third is the clinical development phase. And in the intro course several weeks ago, I showed you this particular slide. And what it is attempting to do is give you a schema of the evolution of drugs to the point of their use in clinical practice. So you will see on this slide, over in the top left-hand part of the slide, is, sorry, let me go back here, is the preclinical testing. And this includes the in vitro studies, which is the drug discovery phase. And then the second piece is the animal testing. And then the interface with the FDA, or the regulatory authority in the country in which you are listening from, is the first interplay before you actually go into humans. And then sequential phases of drug development in humans, and then further review by your regulatory authority to either give the green light or not to the drug being used in clinical practice, which is this phase here. And then post-marketing surveillance after the drug has been approved to ensure that it continues to meet the safety and efficacy requirements of the regulatory authorities in your country. So we're going to use this schema throughout the next three sections, 9, 10, and 11, to go into further detail of how we apply the basic principles of pharmacokinetics and pharmacodynamics to this process of drug development. So the drug discovery and the preclinical development process are considered to be somewhat predictable. We use repetitive systems to assess for early drug action and safety signals. And the time range can vary depending on the nature of the drug that you're evaluating, but a ballpark range that I think is reasonable is 1 to 5 years. The human drug development process is much more highly regimented than the preclinical development process. It requires an unbelievable degree of planning and has so many layers of regulatory requirements and complexity that are ever expanding. It's heavily regulated in the US by the Food and Drug Administration, in other countries typically by arms of the government. It's unbelievably expensive and takes a considerable length of time to actually achieve. And I think you'll get the hang of this by the time that we're done here in this section. So section number 9 is going to focus on drug discovery. And drug discovery is this part here. It's the phase of actually discovering the drug, believe it or not. And a lot of this tends to be done in academia before it gets handed over to industry to do the more clinical testing. So this might be based on a whole variety of different activities, which I'll describe in a moment. But the idea is you want to get a molecule. And you want to get a molecule that holds promise for having a biological activity in a biological system whereby benefit would be derived in the human setting. But typically you're talking about cells or model systems, etc. So it's a very, very initial stages of discovery, and it might involve initial stages of synthesis or even purification of the molecule so that you get the cleanest shot at coming up with a viable compound with which you can go into preclinical and clinical testing. So what are the broad ranging or the broad brush approaches to drug discovery? Well, this slide is not intended to be all-inclusive, but it gives you some examples. So the first is through these broad search and screen mechanisms. This could be through chemical libraries. Examples of these would be antibiotics, indomethacin, which is a non-steroidal pain reliever that was discovered after knowing the natural structure of aspirin. Clinical serendipity, I love this one. It's the idea of evaluating a drug for one indication and actually realizing that it has a benefit in another indication. And the example that I've given you here is but one of many, many, is minoxidil. And minoxidil was a drug that was evaluated as a treatment for depression, and incredibly effective antihypertensive drug, but so potent that it led to hypotension and also had some association with depression. So its clinical utility for hypertension is actually pretty limited. But one of the noted side effects for minoxidil was unwanted hair growth or hirsutism, which was more commonly obviously reported in women as opposed to men. Now, taking the side effects of hirsutism, this drug has been refashioned as a topical medication called Rogaine, which is used in the treatment of male pattern baldness. So using the topical application only for the unwanted side effect in the setting of hypertension treatment to the wanted effect in the treatment of male pattern baldness. So this is a lovely example of clinical serendipity. Another approach that you may be familiar with is a molecule design based on the structure of a natural substance. Now, there are so many examples here, but the examples that I've chosen are, in essence, an attempt to try and give you a sense of the beauty of the history of pharmacology. So let's first of all start off with atropine. So atropine is derived from the belladonna plant. It's called deadly nightshade, a great name for a plant. It was by history or by report used by Cleopatra. She rubbed it around her eyes to dilate her pupils in order to make her more beautiful. Atropine dilates the pupil, so it actually has a pharmacologic effect when used as a plant. Of course, atropine has far more deadly effects in terms of heart rate control, but it was from this knowledge of the belladonna plant that our synthetic use of atropine derived today. Aspirin is another lovely story. A reverend in the Deep South in the 1800s, Reverend Stone, discovered that if he lay his patients who had malaria and had high fevers with their malaria against the willow bark tree, that their fevers would resolve. The active moiety in the willow bark was salicylin, which was subsequently discovered to have antipyretic properties and is the substrate in the form of Bayer's aspirin that we use today. Penicillin, I think most of you will know that story as well. So these are the design of currently used synthetic drugs based on the structure of a known structure of a natural substance. The more kind of scientifically exciting component of pharmacology is actually designing drugs known based on the molecular structure of the receptor or the enzyme. And an example that I talked about in our last class was the naturally occurring hormone angiotensin II, which can cause an increase in blood pressure. And we use angiotensin receptor antagonists because we knew what the receptor for angiotensin II was. So by appreciating the structure of that receptor, being able to synthesize molecules that would antagonize the receptor. In the same hormone pathway or enzymatic pathway is another enzyme called renin. And renin is part of the production of angiotensin I, the precursor for angiotensin II. And knowing the structure of the enzyme renin, coming up with a drug that actually inhibits the enzyme by altering the shape of the active site of the renin enzyme. So knowing the molecular structure of the receptor or knowing the structure of the enzyme is a very, very important and common way in which the drug discovery phase might actually be initiated. Endogenously occurring proteins. So our clotting cascade invokes a molecule called tissue plasminogen activator, TPA for short. Tissue plasminogen activator plays a very important role in breaking down clots. In patients who have experienced an acute myocardial infarction as a result of a thrombus formation in their coronary circulation or a stroke in their cerebral circulation, can be given TPA intravenously as a way to try and dissolve the clots. So giving supraphysiologic doses of a naturally occurring protein to actually dissolve the clot is an example of knowledge of an endogenous protein given exogenously to treat very acute medical conditions. Prostaglandin analogs is another good example. Prostaglandin E is important in maintaining the health of the GI epithelium in people who are prone to peptic ulcer disease. So Cytotec, which is a prostaglandin E analog, can be used to try and prevent ulcer formation in people who need non-steroidal anti-inflammatory drugs for pain control. Prostaglandin E analogs are also used as abortifacients in therapeutic abortion. And then there's the final approach of the Me Too approach. And this, from my perspective, is somewhat unimaginative, but maybe necessary from an economic perspective, is the idea of generating a drug which is very similar to a drug that's already on the market. And this typically occurs in the domain of large pharma, different companies competing with each other to get different drugs to the market. So an example here would be Lipitor, which is a torvastatin made by Pfizer, rosuvastatin, which is Crestor made by AstraZeneca, and simvastatin, previously known as Zocor, produced by Merck. These are all medications that are statins in nature and differ ever so slightly from each other and all have the same sort of clinical indication to reduce LDL cholesterol. So knowing the structure of one drug already on the market prompting the development of similar drugs in a Me Too type of approach. Now, the general principles are pretty much the same. Ideally, in an ideal world, what you would love to be able to say that you have done in your approach to drug discovery is decide what your target is, rational identification of a target. And then you take that target and you screen for action against that target. So these are called screening assays for drug action. And then you want to prove your principle in a model system. And typically in a model system is ex vivo or in vitro. So you want to be able to maybe have a visualization of your receptor in a cell membrane model and show that the drug actually binds to the receptor in that cell membrane. Or maybe an enzymatic kinetic system showing that the drug actually either inhibits or activates, less rarely, the enzyme in that model system. And typically what happens in the real world is you're not just working on one molecule. So very often from a biochemical or a chemical perspective, you can tweak these models, add a hydroxyl group, add a double bond, use a stereoisomer, modifications of the fundamental structure of a single molecule in the hope that it might alter, for example, the bioavailability, the hydrophobicity, the lipophilicity of the drug, or that its affinity for a particular receptor may be ever so modified by changing its chemical structure. And ultimately what you want to end up with is one or a few lead compounds that hold most promise at the end of the ex vivo evaluation of this drug discovery phase. And I will tell you that the drug discovery phase is an ever-expanding domain. We are eternally in pursuit of ways in which we can assess drug action outside of animals and outside of humans for obvious ethical reasons. So this concept of coming up with lead compounds for preclinical development is very important and actually also very exciting. And your ultimate goal is to be able to choose a molecule for future clinical development. And for most examples that I've given you previously, it is the selection of one molecule that goes into the human drug development stages. So after that drug discovery, or sometimes contemporaneously with that drug discovery phase, is the initiation of the preclinical drug development phase. And this is also referred to as the animal testing phase. And as you'll see on this schema, you have short-term animal testing and long-term animal testing, and it is not at all uncommon for the short-term animal testing not to be completed before the human testing begins, and it is common that the long-term animal testing is being conducted at the same time as the first stages of human development actually occur. So there is a significant degree of overlap there. So let's talk for a minute about preclinical drug development. And the overarching theme of preclinical drug development is you want to walk away from this stage of drug development with a sense of organ system specificity. You want to have a sense of the safety profile, even if it's in an animal model. You want to get this spectrum of safety that you can anticipate and design your human protocol based on knowing that information. You want to apply your knowledge about pharmacokinetics and try and extrapolate out what the metabolic profile of the drug is in the animals that you've chosen to test. And at the same time, you have no idea or a limited idea as to how your drug is going to behave in the animal testing. So you want to be able to stabilize your drug, whether that means packaging it as a prodrug or whether it means purifying the drug to alter the PK profile or the PD profile. This is commonly entertained at this preclinical phase of drug development. So if you ask yourself, what is the purpose of preclinical drug development? The purpose is to determine the safety margin between the efficacious dose and the toxic dose. So let's return for a moment to our previous class. We talked about the therapeutic index, that margin between the EC50 and the LD50. Here we're actually even narrowing it down a little bit further. We want to know what is the maximum tolerated dose, i.e. the dose before which toxicity is seen. Then we also want to know what's the minimum effective dose. So what is the least amount of drug that we can get away with in order to demonstrate the efficacy that we're looking for. And then we do, even though ethically it does raise significant eyebrows, we do assess for the lethal dose in animals. And this is a requirement in order to be able to understand what that therapeutic index is that we've talked about previously. Then you want to know, where do you want to start in humans? So a second purpose of preclinical drug development is to define a safe starting dose in humans. Now this may sound a little bit crazy to you, but the safe starting dose in humans is somewhere between 1/10 to 1/100th of the no effect dose in animals. And the no effect dose is exactly what the words would imply, the dose at which zero response is seen. So this is an extraordinarily cautious approach to exposing humans to drugs for the first time. Now, it does, of course, have an ethical downside to it, that we're exposing humans to experimentation at doses for which we don't anticipate any response, and that's a conversation for another class. But somewhere between 1/10 to 1/100 of the no effect dose seen in animals. We do this preclinical drug development in addition to characterize the toxicity that is exemplified by the animal model. And you're going to see we do more than one animal model. But what we ultimately want to do is highlight the organs that are at risk for toxicity. And then we take that information into humans, recognizing that we need to have a heightened level of awareness for the organs in the human body that may be at risk for toxicity. And in the process of not only identifying the organs at risk for toxicity, but we want to try and understand the mechanisms responsible for the toxicity. And in preclinical and clinical drug development, we typically refer to these as adverse events. Particularly in human drug development, we talk about them as being adverse events. This is a way in which we measure toxicity. And then to return to the first weeks of the class, we evaluate the PK profile in animals. And just to refresh your memories, which I'm sure I don't need to do, the PK components of drug profiling include routes of administration, absorption, distribution, metabolism, and excretion, AADME. And it includes an estimate of bioavailability, and then harking back to what we said previously, recognizing gender differences in drug response, we incorporate those when necessary. Let's, for example, say in birth control or hormonal-based therapies. Disease states, so volume overload states, patients who have genetic deficiencies of receptors, we create knockout mouse models typically. Drug interaction studies where we give animals cocktails of drugs that would commonly be used in the clinical setting for which the drug is hopefully being intended for, to try and replicate the types of drug interactions that might be seen or expected to be seen in the human clinical studies. So I would really like you to be able to remember what the primary purpose of preclinical drug development is. And for many of you, you may be struggling to gain a degree of equipoise between the scientific need to characterize these aspects of the drug intended for human use with the ethical concerns of the use of animals in clinical trial development for human, ultimately for human use of drugs. So what are the types of animal studies that are done? Well, here we've depicted them as animal safety studies, and yes, they are primarily safety studies, but we also do assess elements of efficacy in terms of mechanism of action. So let's just run through these. And as you will see with each of these, not all of them are required for all types of drugs used in clinical medicine. So for example, all studies will require acute toxicity. And acute toxicity is usually defined as toxicity in response to single dosing. And you use incremental dosing in cohorts or groups of animals to demonstrate the toxicity. Subacute toxicity and chronic toxicity are going to be required depending on what the clinical use for the drug might be. So for example, there is a drug called diflucan, and it is used for the treatment of candidiasis. It is only one single dose. Acute toxicity studies will be required to demonstrate the safety and efficacy of that particular approach in animals. In contrast, the development of a novel antihypertensive drug, which will be used over extended periods of time in patients with the inexorable diagnosis of hypertension, will need acute toxicity, subacute toxicity, and chronic toxicity. Then in addition, we need to consider other types of safety assessment. These include teratogenicity, the potential impact of the drug in the event that the person taking the medication becomes pregnant, or that the partner of the patient who is taking the drug becomes pregnant. Carcinogenicity studies, particularly important if the drug is considered for longer term use, and mutagenicity studies. So we will go through some of these in more detail. So what is the primary purpose of acute toxicity? Well, here I'm going to introduce some terms to you. The primary purposes are the following: to determine the no-dose effect. Remember, this is the dose that is used to calculate the starting dose in humans, the no-dose effect. The maximum tolerated dose, as we said, the dose at which no toxicity is seen, and the lethal dose at which, and we use the LD50, the dose at which 50% of the animals tested die. We also use the acute toxicity studies to study the intended route of administration. Typically, the acute toxicity studies are conducted in two different species, commonly a rodent species and a rabbit or a dog, and two different routes of administration. And the primary reason for the two different routes of administration is, in order to calculate the bioavailability, you need the comparison of the IV administration route. Remember back to that second class, bioavailability is the comparison of the amount of drug that reaches the systemic circulation following oral delivery in comparison to following IV delivery. So we need to study two routes of administration in order to be able to determine bioavailability. Subacute toxicity extends it out a little bit further. Regulatory agencies will differ depending on which country you are operating in, but they typically require, again, that two separate species be studied. Same deal, one usually has to be a non-rodent. And the duration of testing is actually quite variable. It can be as short as 2 weeks, and it can be as long as several months. And it is predicated by the clinical indication for which you plan to use the drug. So let's say, for example, there is an antifungal agent called Lamisil, and the typical duration of action of this drug is, the typical duration of treatment of this drug is 6 weeks. So the subacute toxicity testing for this particular study might last out to several months, whereas a 3-day or a 5-day course of an antibiotic, the testing may be as short as 2 weeks. Here, 3 dose levels are ideally studied: the safe dose, several multiples of the projected starting dose in humans, the dose causing minimal toxicities that are reversible. So where's your level of acceptable toxicity? And then the dose causing frank toxicity. And ultimately, you want to be using the safe dose, several multiples of the projected starting dose, but you want to minimize the toxicity, and you most definitely want to avoid a dose that causes frank toxicity. And here, the subacute toxicity is really trying to hone in on the concept of risk assessment and therapeutic index. And the duration of treatment is to match the intended clinical use, as I said previously. Here you're doing your typical evaluation for toxicity. This will include clinical chemistry, such as white blood cell count, or renal function, hepatic function, etc. Hematology, that would include your white blood cell count, your red blood cell count, your platelets. Histopathology, actually sections of tissue looking for histopathologic change suggestive of toxicity. Typically, subacute studies are done in cohorts of animals. So you have your 3 different dosing levels, and you study groups of animals in contiguity. And you would assess plasma and urine levels for whatever levels of toxicity. You would also assess plasma and urine levels for metabolites and the manner in which the drug was excreted. Again, following back to those basic pharmacokinetic principles that we talked about previously. And the subacute toxicity very often are partnered with the acute toxicity, because what you really want to be able to demonstrate is what is the pharmacokinetic profile following single administration of a drug and repeated administration of the drug. Remembering that very often toxicity may be cumulative, that you would actually increase your likelihood of toxicity following repeated exposure to a particular drug. Also, efficacy can be considered to be cumulative, and they would both be pharmacodynamic principles. Chronic toxicity is generally employed where long-term administration is intended for clinical use. So antihypertensive drugs, cholesterol-lowering drugs, drugs that lower blood sugar, drugs that are used to prevent asthma, drugs are used to try and control CD4 count in HIV-infected individuals. So drugs that are considered to be for the management of long-term, as yet incurable diseases. The chronic toxicity studies are usually conducted for a duration of time that's more than 6 months. These are also done in 2 species, one of which would be a non-rodent, and the rabbit is most commonly employed for the chronic toxicity studies. The same outcomes, exactly the same outcomes as the subacute toxicity. You're trying to do, the purpose is the same. You're trying to do the same thing. You want to characterize the PK profile in the chronic setting. You want to try and get a better sense of the mechanism of drug action. You want to try and get a better sense of the dose response, particularly as it relates to toxicity. And importantly, the chronic toxicity studies don't absolutely have to be completed by the time you go into humans for the first time. The metabolism studies that are conducted at most stages include all of the testing to exemplify the PK principles. So route of administration, absorption, bioavailability, metabolism, rates of elimination. We use the area under the curve to try and extrapolate not only the PK profile, but also the pharmacodynamic or efficacy profile. We also want to know what the effect of changes in the host that can affect PK, just like the acute toxicity study. So does increasing age of the animal under investigation alter the PK profile? If we ligate a renal artery in the animal that's under testing and impose renal dysfunction, does the handling of the drug alter significantly? Is toxicity manifest in an animal who has been devised to replicate renal dysfunction? Is there any alteration based on gender, or that can be defined as a function of gender, and drug interactions like we said previously? In general, the other preclinical testing is typically 24 months in nature, and this is considered to be equivalent to a lifetime in an animal, which is a somewhat surprising equivalence to draw. It's also usually done in 2 species, a rodent and a larger animal. In this example, it's given a rabbit. However, some studies might actually involve a primate, such as a baboon. Human studies can also commence prior to having these data. So the teratogenicity data is essentially the data that affects reproduction. And the sorts of questions that we're interested here is, does the drug alter mating behavior? Does it alter reproduction potential? Does it affect parturition, which is delivery of the offspring? Does it affect progeny? In other words, are the offspring adversely affected by exposure in the mother or in the father? Birth defects. Are there any significant birth defects as a result of being exposed to the drug, and is postnatal development of the progeny adversely affected? So these are all manifestations of teratogenicity, which are more relevant to humans the higher up the animal model that you decide to evaluate. Carcinogenicity testing is typically done in the pathology lab. So both gross pathology, anatomic pathology, and histopathologic pathology, looking down a microscope, to look to see of any subclinical, in other words, the animal doesn't manifest the toxicity, but subclinical manifestations of toxicity if you look at it closely enough in tissue section. This is particularly done if the drug is intended for prolonged use. So again, the same examples: antihypertensive drugs, drugs for patients who are HIV positive, oral hypoglycemic agents for diabetic patients. And then finally, mutagenicity. And this tries to get at the point of genetic stability. And here there is an effort to try and identify mutations in either cell cultures or bacteria model systems to try and extrapolate the potential of mutagenicity in humans should it get that far. So in this particular slide, you can see that you have completed all of these 2 phases of testing. And the entire process for which that actually takes place can be variable. The limitations of preclinical drug testing warrant attention. First of all, animal testing is expensive. It takes a long time. Very often it has to be replicated. As a result, large numbers of animals are necessary, and this raises significant ethical concerns, as mentioned previously. There is an overwhelming drive to limit the total number of animals that are exposed to any research procedures on a national as well as international level. And there is a real drive through the development of consortia across large pharma houses together with academia as well as regulatory bodies to try and develop novel cell and tissue culture techniques that would enable safety assessment of drugs and ideally would obviate the need for animal testing in the preclinical drug testing phase. We're a long way off from getting to this point, but it is certainly an initiative that has a lot of enthusiasm behind it. The third point to consider is that extrapolation of toxicity and efficacy may not always apply to humans. And this is really important. It is the human model that counts. And even though we can go higher up in the animal kingdom and try and get as close to humans, for example, with baboons as we possibly can, certainly the rodent model does not mirror human biological systems. So we may be able to identify what an enzyme target might be or what a receptor target might be, but the ultimate toxicity in humans requires ultimate evaluation in humans. So even though it gets to the point of entering into humans the first time, we don't have a total handle on the efficacy and toxicity of the molecule that we intend to use. And most importantly, because the total numbers of animals that we use in preclinical testing is ultimately quite small, rare adverse effects will likely not be manifest. And therefore, we have to have a very heightened level of awareness for the potential of toxicity that has not heretofore been made apparent in the preclinical phases of drug development. So we'll end this particular section and move on to the final section for our next class, which will be part 3, clinical drug development, as our final section to this course. Thank you for being with me in class today. I hope you enjoyed learning this material, and I look forward to seeing you back in class again shortly.